Welcome back for more AP Biology. Today we're going to be talking about how the nervous system functions, which means in the last video I kind of lied a little bit when I said I won't be making videos about the nervous system. I'm not making them for the immune response, the defense mechanisms, and for how hormones function, because those were presentations done in class. So you all did that teaching, not me. So what we're going to look at is the role of facilitated transport and active transport in how neurons do their jobs. And this is something the College Board likes to test on, and there's lots of stuff involved here. So, we need a refresher. How do we move substances in and out of cell? Well, it depends on what the substance is. If we have something that's nonpolar, it just moves right through the cell membrane. Seems easy enough. If it's something that can move through the cell membrane but it needs a little bit of assistance, we use facilitated transport or facilitated diffusion. Well, that's pretty simple. So we just need a hole and things can move back and forth. But there's some things that we need to force the movement of. So we're forcing a change in concentration, namely to go from a lower to a higher concentration, and that we call active transport, and we've dealt with all that stuff before. So if I were to take a cell, in particular something called the sodium-potassium pump, which all cells, or at least all eukaryotic cells will have, what we can notice is this thing would utilize ATP energy, so it's going to put in energy, and it's going to force sodium ions out of cells, and it's going to force potassium ions into cells. Well, why would you do that? What you end up creating is a concentration difference. So there's a high concentration of sodium ions outside of the cell, very low concentration inside the cell. High concentration of potassium ions inside the cell, very low concentration of potassium ions outside of the cell. Which means if you give them the opportunity to move, sodium will diffuse into the cell or influx. Potassium ions will leave the cell or efflux. When you do that, it turns out that we have another little channel called a leak channel, meaning it will allow a slow movement of ions. And this one in particular will allow potassium ions to leak. Okay. So potassium ions are allowed to very slowly leak. Well, where would they go? They would actually leave the cell. They will efflux. One of the things that we don't talk about a lot is whenever you have a positive charge, you must have a negative charge. So whenever we have potassium ions leaving the cell, they will leave behind negative charges. Positive charges and negative charges are attracted to each other, so the result is they want to be attracted. Potassium ion leaves because of concentration, but it doesn't want to leave due to attraction to an opposite charge. What this ends up creating is what we would call a membrane potential. That membrane potential is a number. It's a voltage. It's like a battery. Well, and batteries we can use. We can quantify it using this particular relationship here, referred to as, this is referred to as the Nernst equation, and when you take chemistry, if you haven't taken chemistry yet, you get to calculate membrane or cell potentials using this particular equation. It's not difficult. You just plug numbers in and then you hit a button and you get the answer. What makes it strange is it has something in here called ln. It's not in, but it's ln, which stands for the natural log, which if you don't know what logarithms are, makes the math a little confusing. So, all cells turn out to create this thing called the membrane potential. Every cell is a battery. Not every cell is capable of understanding what to do with the fact that all of the cells are batteries. One of those cells that can utilize the battery is referred to as a neuron. The basic structure of a neuron is you'll have something that we call the cell body, which is where you'll find the nucleus. Then we have these cytoplasmic extensions off of it that we call dendrites, because they look like branches. There'll be one particularly long extension, one very long branch that we call the axon because it looks like an axis or a rotor. And then that'll terminate in like a whole bunch of little hair filament-y things that call axonal terminals or terminal buttons or something along those lines. You'll also notice here that we have layers of stuff around the axon, which they call Schwann cells, and they create a layer of fat called myelin, with little itty-bitty spaces of the axon showing up called nodes. Okie dokie. Well, what's the point of any of that? It turns out using that membrane potential creates something that we call an action potential, or it's a signal. Well, what's the signal? The signal is basically this. Lights out, lights on. Lights out, lights on. Well, that's silly and dumb, but we could send information this way. Lights on, lights off. We can do this. How does that work? Well, basically, if you think about it, it's like computer coding. Computer coding uses, in part, a binary system. We're ignoring like hexadecimal and stuff like that. But binary is a 0 and a 1. So as long as you can make combinations of zeros and ones, we can say anything we want. But that's all this turns out to be. The resting membrane potential turns out to have a negative value inside of the cell. So if I were to make sodium ions dump into the cell, I can make the inside of the cell become positive. And if I can get potassium ions to leave the cell, it becomes negative again. So all we're going is from a negative to a positive to a negative. That's like saying 0 and 1 and 0 and 1 and 0 and 1, which is binary, and we can communicate that way. So what is the action potential? Action potential starts off with, if you look down here in the picture, so where it says number 1, we're at rest. Nothing's happening. There's going to be some type of signal, probably due to cell signaling. That's going to cause sodium ions to start to leak inside of the cell. As sodium ions leak inside of the cell, it's going to make that negative value become greater, head towards 0, because if you add positives to a negative, we become less negative. You will reach a point at which you can't maintain this anymore, and it's going to open up a type of gate, gates open and close, called a sodium ion voltage gate. We open up a sodium ion voltage gate, we change the state of the charges, which will change the structure of the protein, which will open it up. The result of that is sodium ions dump into the cell. Sodium ions dump into the cell becomes positive. Well, it once was what we would call polarized, but I'm undoing that. So we would call that being depolarized. And that is on that middle graph, number 3. Well, sodium ions are going to rush in, but that rush in is going to make the voltage shift such that the sodium ion voltage gates will close on them. They can't handle the change, they close. But that voltage shift is enough to make potassium ion voltage gates open up. And what will that do? Potassium ions will leave the cell. They will efflux. So we have a positive inside of the cell, and we're getting rid of all the positive charges. And if you get rid of enough positive charges from a positive cell, eventually you get a negative cell. The result is we got our polarization back, or we are now repolarized. The catch with all of this is you'll overshoot it, because it turns out there's a delay in closing the potassium voltage gates once we repolarize. And the result of that is what it says is number 5 there, which is you kind of overshot it a little bit. So we have a state of being hyperpolarized. Well, what does hyperpolarized mean? If you're hyperpolarized, it's going to be harder to become depolarized, and the result is we can actually have a direction to this. We can't just suddenly say, nope, just kidding, and let's switch and go backwards. That hyperpolarization means we have to wait. We have a pausing period. So if I were to make the graph, this is what it turns out to look like. So we have nothing going on. We add a stimulus. We leak in sodium ions. That triggers a sodium voltage gate. Sodium ions influx. We have depolarization. That will close sodium voltage gates, open potassium voltage gates. Potassium ions efflux. As they leave, they overshoot where they should have stopped. That creates that hyperpolarization state when those potassium ion gates finally close. And that causes a pause, a mandatory rest. Fancy way of saying that is refractory. And then we can go back to normal. How do you go back to normal? Sodium ion or sodium potassium pump and potassium leak channel. But if I'm going to make a neuron, neurons turn out to function, meaning they move information back and forth. So how is that possible? Well, it turns out if I have one area, yay, this new mouse. So if I take this blue area and I have this area being depolarized, so sodium ions are rushing in, some of these positive charges could wander over to the left, or pardon me, to the right. And if that's kind of like saying we have a sodium ion leak. And if that happens, that might be just enough to reach threshold, and then it will depolarize. Well, if we have enough of these dumping into the second segment, that could be enough to make the area just to the right of it feel like it's going to hit threshold, and then that will depolarize. And the result is we have a one-directional flow of this action potential. After the depolarization, you get a repolarization, but it turns out a depolarization will trigger another depolarization, which will trigger another depolarization. And this is going to continue until we reach the end of the neuron. Once we reach the end of the neuron, the depolarizations are actually going to trigger not another depolarization, but the release of chemicals. Those chemicals are found in little bubbles or vesicles, and the name of the chemical is called a neurotransmitter. They will fuse with the cell membrane, will have exocytosis, and those chemicals will dump into a space that we call the synaptic cleft or the synapse. It will leave the neuron, the presynaptic membrane, and it will bind to the opposite side, whatever it turns out to be, which we can call the postsynaptic membrane. Well, these neurotransmitters are signals, so they'll bind a receptor and that will cause something to happen. What's going to be that thing? It depends on what the postsynaptic membrane could be. It could cause another depolarization. It could cause a stoppage of depolarizations. It could cause a muscle contraction. It could cause hormones to get released. It could cause you to make saliva or stop making saliva. It really depends on what's on the other side. It turns out not all cells have the exact same type of synapses. There are things that we call chemical synapses, which is we have to deal with neurotransmitters. But it's also possible for what we call an electrical synapse, which is we have an actual connection that allows the depolarization to move from one cell to the next. Plants, it turns out, like to use electrical synapses. Our heart, it turns out, uses electrical synapses. But our brain uses chemical synapses. Well, what happens if you put a whole bunch of these things together? If we have large collections of all of these parts, you end up getting fights, meaning you'll have some things that say, the answer is depolarize. We'll have some signals that say don't depolarize. The result is we're getting lots of inputs and we need to process it to get one output. We can call that a computer, or we can call it a brain. It doesn't matter what we're talking about. We have a whole bunch of roots that are all connected to each other from a couple million plants that will function like a brain, because it's how are all these interactions being processed. And as long as we have lots of input, we have to process it to get an output. We're dealing with brains. With us, it's trapped in one spot, but it doesn't need to be trapped in one spot. It could be a series of connections underneath the soil. There's nothing to stop that from being a brain. What do we, animals, do with this? We use this to create things that we call reflexes, which is the gross, meaning big, version of cell signaling. Meaning, if I were to go and whack your knee, I'm going to give you a signal, you're going to process it in your spinal cord, and then I'm going to spit out a response, which is self-signaling. It's signal, process, respond. That's all it turns out to be.